Hello, in this video we will discuss about the solid DNA sequencing method. This basically is the based off of Polony sequencing developed by George M. Church and at Harvard University. So remember, this is the different as compared to the Sanger method, DNA sequencing, and the Maxam Gilbert. So this is a different and very cost effective. So in this way, it is basically is the less cost because 0.13 dollar per million bases can be generated, and has trouble with palindromic sequences. So the palindromic sequences which that will create the trouble. What is the palindromic sequences? Let's begin to understand. So what is the palindromic sequences? First of all, we should need to understand that. So the palindromic sequence is a sequence of DNA, a read in the 5' to 3' whose complement is the same when read in the 5' to 3' direction. So that means same complement. Basically, when we will read from 5' to 3', if we will read the 5' to 3', that is same, that is complementary. While the non-palindromic mean when we will read from 5' to 3', and the other complementary when we will read 5' to 3', basically different to each other, not a similar, so not a complementary. So remember, this is a non-palindromic. So next, we will also learn about what is the basically is the issue with palindromic sequences. So first of all, we should need to understand the solid processes. So in this way, the sample preparation first. After the sample preparation, the amplification via PCR. So the PCR required for the amplification, but the different PCR type. So the ligation reaction and imaging, basically is the data collection. After the data analysis is the next step. So first of all, let's begin to understand what is the fragmentation, basically the genomic DNA. So in this way, here you can see this is the cell. When we will separate the cell, this contained DNA will be segmented. So it can be a smaller segment. Creation is known as the fragmentation because it is produce the fragments. So the type of fragmentation is the different, the nebulization. In this way, the compressed nitrogen, which that will be inserted through a small hole to the target to the DNA for the cutting and to fragment creation is known as the fragmentation. So all this happening in this here you can understand in this diagram. So anyhow, if let's we will discuss the sonification. The sonification means the ultrasonic waves. The ultrasonic waves, the sound waves, which basically create the sample resonance vibration. The resonance vibration is basically the ultrasonic waves used to create gas bubbles in sample. And shear DNA by resonance vibration will lead to fragmentation, same. And the third step can be the digestion. The digestion is basically use the kit of the containing enzyme, restriction enzymes, which that will put onto the sample containing cells, will target to the DNA for cleavage, is known as the cleavage DNA to produce the fragmentation. So let's begin to understand the ligation of adapter sequences. So we can use two type of adapter, P1 and P2. So the adapter means it will be bind with the 5' site containing P1 adapter, while the 3' containing DNA sequence containing P2 adapter. It is basically the ligation. So the ligation of adapter through a ligase enzyme use After the using, the attachment will be occur. This attachment is 5' containing P1 adapter, while the 3' containing P2 adapter. So this is the purple color is basically the DNA segment. After the fragmentation, we can use, and after this the fragmentation, the adapter will be attached, and the sequence fragment with P1 and P2 adapters attached. And in this way, the attachment from 5' to 3' is the specific for the P1 and P2 adapter. So that is why it will be bind with the 5' side and 3' side. So after this, the complete the ligation of adapter sequences, the PCR, the amplification of fragment. So the amplification of the fragment required the PCR. PCR means the polymerase chain reaction. This polymerase chain reaction is basically required the solid DNA amplification of fragments, which that contain a reagents. Reagents basically the used for the solid DNA amplification. Amplification means the number of the segment will be increased by the reagents using the template strand from PCR, and the primers to start the reaction. The specific primer used for the amplification of specific DNA segment, and the 8 nucleotide long probes with dye, basically with color producing. So 8 nucleotide long probes, and the ligase, a ligase to add and probes to primer. So in this way, basically is the probe is basically is the uh, 8 nucleotide containing sequence is containing the color, which that is used after the amplification produce color when bind with the template strand, the as a sample the segment of the DNA. So in this way, here is the PCR bead we are using, which that's containing a P1 adapter. So the bead cover to segment corresponding to P1 adapter. So I am here not drawing, but remember the P1 adapter is attached with a bead. So after this, that is why the segment through a 5' side can be bind. So remember, after the binding, this bead basically containing each fragment bind to its own beads. So each bead has a unique nucleotide sequence. So each bead containing a unique nucleotide sequence. So that is why remember that things. So a unique nucleotide containing sequence, which that will be amplified when we will apply the primer for specific primer. So the each fragment binds to its own beads. So each bead has a unique nucleotide sequence. So that is why here is basically after the binding the PCR bead, million of copies of template strand on each bead containing here. So in this way, the called the polonies, which that contain a polymerase colonies, which that is used after the binding of the primer to use annealing to produce amplification of the multiple type of segments containing a different sequence. So the millions of copies of template strand on each bead containing. That is why it is the major important and not all beads fragment bounds, but the other bound. So what about the empty beads? Empty bead is the different, which that's contain a polystyrene bead coated with P2 adapter protein 2 adapter 2 segment, and which that is contain polystyrene, while here you can see this the non-bound, basically the non-bound beads, and this is the bound beads, which that will be bound through a P1 adapter, while the polystyrene bead coated with P2 adapter. In this way, it will be bind via a P2 side. So that is why the 3' end can be bind the segment. So in this way, the P1 containing PCR beads will bind also to form this collection. And in this way, the PCR bead no bound to large bead, and that is what here is the non-bound bead and the bound bead. So that is why, what is the significance of that? Basically it will use for the more dominancy. So how it's possible, let's here we will discuss. So then centrifuge out the bead once. So here is you can see the centrifugation will be occur the bead ones. How to separate the lighter beads and the denser beads. So the lighter beads either containing the P2 adapter or not, it's okay, but remember the P2 adapter containing polystyrene bead while the P1 adapter containing a PCR beads. So that is why the both beads is important. When bound with the both P1 and P2 adapter via a polystyrene bead and the PCR bead. So in this way, smaller PCR bead will be dense, while the on top the supernatant which that's containing polystyrene structure are less dense and then group empty beads because they are less spread out. So in this way, it will be moved out during the centrifugation of the sample. So the dense PCR beads will be down while the complete 2 adapter side, the P1 and P2 containing complete ligation of adapter sequence, which that will be moved upward due to the lighter. And in this way, we can attach to glass slide for analysis. So remember, I am again repeating that the polystyrene bead is very important for the P2 adapter binding to the segment of the DNA, while the PCR bead no bound to large bead due to the P1 adapter. So remember, the P1 adapter will bind with the PCR bead. So the ligation and imaging, the mechanism, the ligation chemistry we will discuss first. So in this way here the primer bind to template strand first. And the second is the probe hybridization and ligation. And the third one is the fluorescence, the measured. And four is the dye end and nucleotide cleaved. Step 1 to 4 the repeated 6 times. So the sixth time step 1 to 4 will repeat to produce more and amplification to produce a color, the inflorescence. So remember, dye end and nucleotide cleaved. So that step 1 to 4 repeated 6 times. So remember the process completed 5 times. Each time primer is offset by 1 base. So remember the offset by 1 base. We will discuss the whole mechanism. So remember, first of all we should need to understand, let's begin to understand the anatomy of probe. So what is the probe? Probe is used for the producing of coloring. So when coloring detection means our DNA, our specific DNA will be amplified. So in this way, the two actual bases, each dinucleotides permutation corresponds to a dye color: red, green, blue, yellow, orange, and 60 positive dinucleotide permutation. While on the other hand, this is the universal basis, bind to any of the 4 nucleotides. So remember, it can be bind with any 4 nucleotide. While this is the universal basis, the pink color I am showing, the with fluorescent dye. This is the fluorescent dye. What that means? Measure for fluorescence and cleaved in each cycle. So attached probe on only 5 nucleotide side. So in this way you can understand this is the anatomy of probe. So this is the fluorescent dye which that will produce color corresponding to 4 dinucleotide possibilities. 4 nucleotide possibilities. So next here we will discuss about the basically is the another way for the ligation, sequencing by ligation. So in this way we will discuss the whole mechanism, ligase, seal, the 5 prime, the nick. Here I am drawing the beads attached to glass slide. So in this way the beads containing a P1 adapter with a segment, while the P2 adapter is the polystyrene bead side. So in the bead anchored, the templates a trend. So in this way, this is the 5' side contain thymine and adenine, the two bases, which that is the oppositely bind two base encoding the adenine with thymine is the oppositely to the 3' side, and ligase seal the 5' the nick. And in this way, the basically the N is the number of the nucleotide will point will encoding, and in this way the bead anchor the templates strand. And after this, the basically is the step 1 here is the bead attached to glass slide first. After this, the step 1 here, which that we had discussed. The step 2 is the fluorescence will produce after the excision, the exciting to produce color. So here you can see this is the 3' and 5', the T side thymine and adenine. So this is the excite to produce the fluorescence, the color. So after this here is the fluorescence cleaved off, the third step. So in this way here you can see this is the after the cleave off to the produce the fluorophore in the third step. The fluorophore cleaved off to produce the color, and that color will can be detected. After this here is the 3' side containing adenine and thymine, and the nucleotide more nucleotide and to the thymine to the guanine bind side, and the thymine to the guanine again. And this is the extension we will discuss in the next time, next diagram. So this is the recording of the fluorophore cleaved off after to record the color, while this is you can understand this is the basically is the 5' here you can see the 3' to adenine thymine. So this is, let's begin to understand the step 4. So that's in step 4, the basically the 3' sides containing adenine, thymine and nucleotide more adding to the thymine with guanine, thymine guanine, which that is the antiparallel, and the in front of the adenine with cytosine, and to produce the different type of color. And this is the extended strand with a partial sequence read by 2 base encoding. So the 2 base encoding will read the color, and partial sequence read by the 2 base encoding. So in this way production of color which that can be detected. So in this way that is known as the basically real time PCR. So it is show the color also. So it is the basically the type of look like the real time PCR. So remember, so this is the after the free 5' phosphate available for next round of probe attachment after the extend sequence melted away. So this sequence will be melted away to bind again after the new primer will be bind. Before the new primer, the primer reset and repeating the 2 base encoding process. So in this way primer can be bind, while in this case the step 5 basically again can be repetition will produce more color, the different type of color. So the different segment can produce the different type of color. So the primer can bind after this, the step 6, and the new primer will bind will annealing the primer reset and repeating the 5 base encoding process. So anyhow, this let's begin to understand again the simple diagram to understand the probe attachment. So how the probe can be attached? The probe is that nucleotide sequence which that contain fluorophore. After the reading, after the binding will cleaved to produce color. So this is different type of probes on the basis of specific sequence will bind the primer corresponding to P1 side, the P1 adapter side. So in this way, here is the ligase bind, annealed, probe by joining backbone. So it will be joining the backbone, the ligase enzyme used to primer corresponding to P1 site. After this P1 site, suppose this is contains yellow, specific probe will produce color after. Let's begin to understand this. The different fluorescent fluorescent dyed oligonucleotide 8-mer or mere probe, basically the 8 nucleotide containing probe, when bind with the DNA, that DNA is the template DNA or sample DNA, which that is collected through a sample. So that is known as the sample DNA will bind with the antiparallel strand, the probe. After this probe when bind, what will be happen? So remember, the fragment being sequenced lower energy photon corresponding to dye color leave and activate the detector. So the add detector will be activated after the cleavage corresponding to dye color leaved and activate the detector. So the detector measured and record the fluorescence. After the recording of the fluorescence, the laser excites the fluorescent dye. So the laser excites the fluorescent and dye to produce color, and that color will be record and detector measure and records the fluorescence. So anyhow, this basically the result of one round is not enough. So we can run again and again, but remember the complete mode cycle. 5 cycle can be possible, and 5 cycle is enough for that, and incomplete data will be complete data after the 5 cycle or complete more cycle to incomplete data to complete. So how this is? Basically, this is adenine with guanine, which that's containing adenine with guanine, which that produce the color, the complete Moore cycle. So this is the complete Moore cycle will produce, but we only have 4 fluorescence measurements PCR even fifth base, so that we can 5 cycle create to produce this different type of color, to produce the different type of dye, 2 nucleotide containing segments, which that produce color after the probe binding, because it is probe. So remember, so probe will be separate the two nucleotide containing segment will produce color. So and let's begin to understand the data analysis. So the data analysis mean we will discuss basically is the segment sequence which that produce the more different type of color. Here you can see the different type of color, and this color can be detected, and the first base basically the adenine, cytosine, guanine and thymine, which that we will use to the Punnett scale rule. So this is the bead will bind with the P1 adapter to the thymine side, which that will be bind. Cytosine and guanine, the second base. Basically, the second base, the cytosine and guanine. This is the second base. The first base is the can be the thymine or here, so you can see, and the thymine, cytosine, adenine, and cytosine is the end, and the base a base and a base and a color define the next base in the sequence. So remember, here you can see the basically the, for example, the adenine, which that the adenine show our cytosine to produce the green color. So remember, this green color producing, this is the, you can understand, this is the data analysis to produce more color, so we can identify base and a color define the next base in the sequence. So in this way we can identify, here you can see this is the production of color, so I can match here This is the AC which that is using here. So that is why it is green, because here is the AC side, A into a C to produce the green color. So this is the basically is the function of the base and the production of color and the analysis. So this is the palindromic sequence. A non-palindromic sequence issue with the palindromic sequence, because it is the palindromic sequence will and anneal to itself, creating a local hairpin. So the local hairpin means why? Because here I am drawing the palindromic sequence. It is not a non-palindromic sequence. Contains C, G, G, A, T, A, T, C, C, G, which that will be guanine can be interact with the cytosine to produce the hairpin. For example, this template DNA is inaccessible. So that is a problem. That is the issue with palindromic sequence. Otherwise, it is okay and that is good. And it is basically color producing process to detection either our DNA segments is amplified.